in pursuit of health and wisdom. Sapio with Buck Joffrey.
Welcome back to Sapio with Buck Joffrey. Of course, I am Buck Joffrey, and today I'm going to talk about something that I find absolutely, positively fascinating, and something that you've probably maybe heard of a little bit, but I want to sort of help you to kind of understand what it is and to understand the potential implications for human, health and longevity in the future. And that is gene editing. I know there's different levels of background on this, but let me just start talking and I'll explain it as I go along. Okay, so let's talk about the origins of gene editing, right? So what is gene editing in the first place? As you probably know if you listen to this show and you've got some background from high school biology, you have chromosomes that are, 23 from your, mother, 23 from your father, and that makes 46. And all these chromosomes, they're basically coded with what is called DNA. And those DNA, the way to think about it is just think like a long set of repeating codes, like letters in an alphabet. And there's typically, there's only really four of them. There's A, T, C, G, right? And that's why I'm just giving you the abbreviation for, like, A is adenine, T is thymine, but let's just say A, T, C, G. And so what's interesting is that you get a series of these kinds of codes that ultimately spell out specific genes. And you might have like several thousand of these ACTG things in a row, and that gives you a gene. And that gene ultimately gets read and transcribed into what's known as RNA. And RNA then goes on to be translated into a protein. And a protein ultimately is what creates a certain kind of function in your body. Okay, so we're going to kind of come back to this over and over again. So that hopefully gives you some very basic background. And, for those of you who don't know this stuff at all or haven't had a lot of exposure to it, the idea of gene editing is exactly what it sounds like. Because as you can imagine, when you have these genes, these codes, and you have a mutation, some kind of gene that isn't coded properly in such a manner that it results in a protein that is fully functional. And that is a problem, and it creates disease. So a good example of that would be like sickle cell anemia or cystic fibrosis, where you have essentially you have these genes that are faulty. And so the concept with gene editing is, in theory, could you go in and edit what's gone wrong with those specific genes and turn them into normal genes, and thereby correcting the actual disease that you see in the human being? Okay? The journey of this whole concept of gene editing started back in the 1970s. There were certain types of enzymes that could actually cut DNA at certain areas, but it wasn't really until fairly recently, where there was the advent of something called CRISPR, CRISPR, hyphen Cas9, so CRISPR-Cas9. And that technology has really become a game changer. Now, so CRISPR stands for, not that it really matters, but I will tell you anyway for being complete, CRISPR stands for clustered regularly interspaced short palindromic repeats, okay? And that was discovered in the late 1980s in bacterial genomes, but it wasn't until 2012 that Jennifer Doudna and Emmanuel Charpentier published their groundbreaking paper in the journal Science. And basically what they did is they described how this CRISPR-Cas9 system could be repurposed ultimately for gene editing in any organism. Okay, so this is in a nutshell how CRISPR-Cas9 works. Okay, as I mentioned, DNA is the ultimate code, right? That is the ultimate code that you've got. And typically the way that DNA it expresses itself. It goes through transcription, which basically means DNA gets converted to RNA, specifically based on the code of the DNA. You get a specific code of RNA, and that RNA then is read and turned into a protein. So we talked about that once before. Now, what CRISPR-Cas9 does is it allows you to engineer an RNA code that will match a specific gene, a DNA coded gene on the genome. That helps it to sort of map it. That creates sort of this GPS to find the DNA, the gene itself. Okay? Once it finds that, the Cas9 enzyme actually will go in there and wherever that gene is, literally cut it out. Okay? That's CRISPR-Cas9. So that is how you identify the gene that you want to get rid of and cut it out. CRISPR-Cas9. CRISPR takes the RNA, use it as GPS to find DNA, and it sits on it, and Cas9 cuts it out. Really, really incredible stuff. Okay? So as you can imagine, this has allowed scientists to either, disrupt a gene or, as you can imagine, what we want to do often is insert new genetic material. And this technology has really revolutionized genetics, making gene editing more precise, efficient, accessible. Now, there's obviously a lot more to this. I mean, the insertion techniques themselves, there's a lot of technical issues regarding not only cutting this out, but also placing the new gene in place that you want. And it may not be worth necessarily talking about it right now. It gets pretty technical in terms of the biochemistry. But as you can just suffice it to say, there are ways to make this happen. And scientists are working on making it more and more efficient. So how do you use something like this? And I want to talk about this in terms of a practical use case that is already, that's actually gained FDA clearance. So it's not just a theoretical thing anymore. So it's used in sickle cell anemia. So sickle cell anemia is interesting because what it is basically abnormal hemoglobin. People with sickle cell disease have an abnormal form of hemoglobin called hemoglobin S. If you have one gene for hemoglobin S, but the other gene from the other parent is a normal hemoglobin, you won't get sickle cell disease, okay? In fact, having a mixture of a normal hemoglobin and hemoglobin S will give you a protection against various parasites, specifically malaria. And that is why it is thought that the whole thing evolved in the first place, to give protection against malaria, which is a major killer in certain parts of the world. So the problem is when you get two of these hemoglobin S's, you end up with these situations of what are called sickle cell crises, which can be life-threatening, and it's really related to deoxygenation. So when red blood cells containing hemoglobin S release oxygen, the hemoglobin molecules can stick together, and then they form these long, rigid sort of aggregates or polymers. These polymers cause red blood cells to become crescent-shaped or sickle-shaped, that's where it gets a name from, rather than their normal round shape. And that, in turn, causes what's called vasoocclusion. The sickle cell-shaped cells can get stuck in small blood vessels, blocking blood flow to tissues and organs. You get incredible amounts of pain. You get ischemia, which is you don't get blood flow to the appropriate place that you need to because you've got essentially all these clotted blood cells there. And this blockage leads to reduced oxygen supply and, again, causing severe pain and potential organ damage. And it can, it can be life-threatening. And as you can imagine, there's also these abnormal shapes make these red blood cells more fragile too, and so they can break down prematurely as well. So it's important to understand that disease process because here is a situation where something like gene editing can help. There was a recent approval from the FDA on something. It's a CRISPR-based treatment for sickle cell anemia and also a similar disease called beta thalassemia, which we're not going to get into very much because I need to mostly tell you about this so you can kind of get an understanding of how something like this could help in disease processes. Okay, so how does it work? So it's kind of an interesting approach, actually, because in this case, you're not actually cutting out like any sort of specific gene related to sickle cell anemia. Instead, what you're doing in this situation is you're targeting a gene that normally would stop you from producing fetal hemoglobin, right? Like when we are fetuses, we have a different kind of hemoglobin, which is very efficient. And there's a gene called BCL11A, which basically suppresses that production of fetal hemoglobin. And so the idea here is to boost production of fetal hemoglobin by effectively knocking out that gene and suppressing it. So again, BCL11A is basically a repressor of fetal hemoglobin, which we'll call hemoglobin F. And so effectively, what you're doing, you're using CRISPR to edit the BCL11A gene and reduce its function, thereby making it so that the body then produces more fetal hemoglobin. Now, the net result of this is by reducing that function of that BCLA, you get more fetal hemoglobin, and that can effectively replace the function of adult hemoglobin in carrying oxygen. Okay, so you're probably thinking, if you're following me, hopefully you're following me so far on this, but you're probably thinking, well, yeah, that's great. Now you've got more fetal hemoglobin, but what about all this hemoglobin S that you've got in there? Isn't that still going to cause a problem? Well, remember I told you before, if you have one normal sickle cell trait gene and one abnormal one, you don't get sickle cell anemia. You just get basically protection from parasites and such, right? So basically what you're doing here is you're almost creating the same scenario as what we would call the heterozygote. You're creating competition for oxygen between those with the fetal hemoglobin compared to the sickle cell hemoglobin. So you have the competitive stuff, and also there is a physical barrier that prevents aggregation of those sickle cell hemoglobin molecules so that they don't aggregate and create the sickle cell crisis. Again, you can think about this as effectively turning these people into what we call heterozygotes, because again, homozygotes are people who have two sickle cell disease genes and they get sickle cell disease. But if a person has one normal and one abnormal gene, they're not going to have sickle cell disease. And by increasing the amount of hemoglobin F using this CRISPR technology, you're effectively doing the same thing. So making it so that people don't express sickle cell disease anymore. Really cool stuff. And the outcomes pretty impressive. For sickle cell disease, they did a study, and 29 out of 30 study participants had no pain crisis 12 months post-treatment, which is like a really big deal for these people. So that's how CRISPR can specifically address a current disease that's out there. Now, I've just talked about one disease, and the reason I talked about sickle cell is because it's kind of like one of these things that you talk about in med school a lot, right? It's pretty simple. You got one gene, you're fine. You got one bad gene, you're fine. You're a heterozygote, you're homozygote, you're screwed. You've got sickle cell disease, and there's a lot of diseases out there like that. And so there's a number of other stuff that's being targeted. In 2019, there was a study published in Nature that demonstrated the potential of CRISPR to treat something called Leber congenital amaurosis, which is a form of inherited blindness. And researchers were able to basically edit the genes in retinal cells and restore vision in animal models. So it's a really big deal. Another exciting area might be in cancer treatment. The scientists are looking at how CRISPR can be used to modify immune cells to better target and destroy cancer cells. So, really your ability to fight cancer is probably the best chance you got is your own immune system. So trying to create this super powerful immune system using CRISPR is a really interesting strategy. And in 2020, there was a study published in Science reporting the successful use of CRISPR-edited T cells to treat patients with refractory cancer. And again, that's showing, some very promising results there. And you can just imagine how many other diseases you could potentially look at here. I mean, it's being explored as a potential to combat age-related diseases, Parkinson's disease. We've talked here about some of the genes that may be involved in increasing risk of Alzheimer's disease. Specifically, when you talk about like ApoE variants, we know that the ApoE4 variant significantly increases the risk of Alzheimer's disease. So if you have somebody with, 2 ApoE4 alleles, and they have this, 12-fold risk in Alzheimer's disease, it may be the case that you could potentially use some of the gene editing techniques to, again, reduce the risk of developing Alzheimer's in the first place. So again, this is an area that, has really got like endless possibilities to it in terms of treating disease. As you can imagine, again, there are various diseases that increase the risk of cardiovascular disease, like familial hypercholesteremia, which has a few different variants. That's another thing that could be targeted. I would not be surprised if that happens in the future. If you start thinking about where gene editing could really lead, it gets really, really interesting and exciting. We talk on this show all the time about longevity in specific. So let's dive into some of the potential applications of gene editing and CRISPR in the field of human longevity, right? So there's one area of research that focuses on targeting genes associated with aging with a gene known as FOXO3. So you can look this up. It is a gene that's linked to longevity in various studies. You see it in very high frequency in centenarians. And researchers right now are investigating whether enhancing the expression of the gene through CRISPR could extend lifespan and potentially improve health span as well. It's probably important for me to also explain there's another variant of CRISPR called CRISPR-A, right? And what CRISPR-A is interesting because it still uses the same GPS mechanism, right, to identify the gene. So now you're going over to use the RNA to find the FOXO3 gene on the genome, and you're planting yourself there. But now, instead of cutting that gene out, CRISPR-A, what it allows you to do is use the transcription factors to basically amplify that gene. So basically pump out a bunch of extra genes that you're targeting. So that's another way of gene editing. That's CRISPR-A. So again, CRISPR-A is not for cutting the gene out, but it's actually to find a gene that you want to go in there and to basically amplify it. And so that's the idea behind something like how you would use that to amplify the expression of FOXO3. And as you can imagine, there's similar things there for some of the other pathways that we know. Specifically, we talk about sirtuins. The various sirtuins obviously have a significant role, at least in the scientific literature, in potentially increasing longevity. For example, if SIRT1 seems to have a critical role in maintaining genomic stability and metabolic health. SIRT6 also seems to have a major role in metabolic regulation and longevity as well. So again, there has been some interest in using CRISPR-A, as we talked about, for activating or, sort of amplifying the expression of these kinds of longevity genes. So anyway, really interesting stuff when you start thinking about what the possibilities here are. Another potential role of CRISPR-based technology, again, really super fascinating thing, is its potential role in reversing epigenetic age. So we've talked many times about this concept of epigenetic age, and basically it goes along with David Sinclair's concept of aging, which is that over time you've got your genome, and then you have your epigenome, which is the various DNA that sits in between the genes that really determines ultimately which genes turn on and which turns off. So the epigenome really ultimately determines, what's the difference between a brain cell and a liver cell and stuff like that. And as we age, the epigenome appears to get these various chemical changes to them. And those chemical changes, they could be like DNA methylations, so on and so forth. And those fundamentally change how our genes are expressed. And David Sinclair has postulated, and I think is probably the leading theory on aging right now, that it is that epigenetic aging that ultimately results in the thing that we know as aging. And so there's been some research specifically looking at the ability of CRISPR systems to actually reverse these types of chemical changes on the epigenome. So, for example, instead of using the traditional CRISPR-Cas9, again, that nuclease that cuts the DNA, they're using inactive or dead Cas9 that can still bind to the DNA sequences but doesn't cut the DNA. But in this case, the Cas9 is fused with enzymes that can potentially modify these epigenetic marks. And those could include, again, DNA methyltransferases, demethylases, in other words, removing DNA methylation. Anyway, we don't need to get into the biochemistry here necessarily, but all these changes that are made to the epigenome, the thought is that you can create a CRISPR system that can go in on those areas that have all of this epigenetic changes that cause epigenetic age and actually reverse them. In terms of longevity, another thing that we often talk about is cellular senescence. Basically, there is one of the things that is a hallmark of aging is an increase in what are called senescent cells. Senescent cells are essentially zombie cells, right? They're not really doing anything. They're kind of like non-functional, but they don't die. And the problem with that is they also release various types of chemicals around them that turn other cells into zombie cells or senescent cells, and are thought to potentially play a role, as you can imagine, in various cancer development. So there is a protein that appears to be encoded by a gene known as CDKN2A, and that appears to be a key regulator of cellular senescence, basically increasing the expression of this gene, CDKN2A, appears to increase cellular senescence and aging. So again, if you can imagine with gene editing, if you can potentially target that gene and downregulate it, then you would get less cellular senescence. So again, hopefully you're getting the idea. There's just all these different kinds of these different strategies that you can use once you can actually play with the DNA, edit it, and so on. And we're really just at the beginning of this stuff. So it's pretty fascinating. And I think it's going to continue to play a huge role in human longevity. Other areas which we won't necessarily even talk about because we've been talking about, as you can imagine, is, the editing of mitochondrial DNA. Mitochondrial DNA does tend to get damaged a lot because it is right in the middle of all of these cellular activities and oxidations and so on. and that seems to be a major player in aging. There's also telomere modification. There's CRISPR-based approaches that are being explored to modify telomere length, which has been associated with cellular aging. Bottom line is, this is a super exciting area. I am just completely fascinated by it. I think it's going to be huge in the future. And my purpose here is simply to just kind of give you a starting point, an introduction, because I don't think a lot of people really know. You hear about gene editing. You might have even heard the words CRISPR, but you may not know exactly what it is. It's real. It's I think it's going to be pretty transformative. You put that together with artificial intelligence, and I mean it really could be key to some of the biggest changes we see in human health and longevity in the future. But anyway, that is it for me this week on Sapio. This is Buck Joffrey. Hope you enjoyed the show, and I'll be back next week.
Thanks for listening to Sapio with Buck Joffrey. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe. Until next time, this is Buck Joffrey for Sapio with Buck Joffrey.